And that also solves we were always scratching our heads before we found this crucial intermediate of lipid peroxidation products, how a signal that's short-lived, such as reactive oxygen species, could reach the electrical impulse generating machinery in the cell membrane when it originates in the mitochondria. And the answer, of course, is that it's not the short-lived reactive oxygen species that serves as a direct signal at the plasma membrane, but the lipid peroxidation products that are produced as a result of the electron leak.
Got it. So the reactive oxygen species are so ephemeral that they don't last long enough that they can kind of be detected and counted and kept track of, but these lipid peroxidation products do. So on the one hand, they're a good signal to listen to because they're nasty. They're clearly indicating that like a lot of metabolism has happened and we need to know that, but they're not so ephemeral that you can't keep track of them.
Yeah.
Interesting. Okay. So just to say it explicitly for people, normally when we think about memory, we often think about, the connections between neurons and things like this, but we're talking about a system here that's all within one cell. This is one cell that's keeping track of its basically its metabolic history by converting this analog signal into a digital signal.
Right. And I mean, the memory, so memory obviously has a neurobiological and a psychological sense, but it also has an engineering sense in computer science. And the memory that's in our computers and in our cell phones, known as DRAM or dynamic random access memory, that was invented in 1969 by Bob Dennard, who worked at IBM Research in the Hudson Valley. And if you look at his patent, the architecture of an elementary memory unit that can hold one bit of information precisely recapitulates the architecture of the ion channel that we've studied. So Dennard's electronic memory is a two-component system. It contains a capacitor that stores one bit in the form of an electrical charge, and then a gate, an access transistor that determines, depending on its voltage, whether you can read or write memory to the capacitor. So you can charge or discharge the capacitor. In our case, the memory unit is that ion channel subunit that flips from a reduced to an oxidized state, depending on the availability of lipid peroxidation products. And whether it can be read out is also determined by the voltage across the plasma membrane.
So human beings dreamed up and built this type of system, and they patented it, and they had no idea about this biology. And it turned out that nature had invented more or less a perfectly analogous system hundreds of millions of years ago.
Yep.
So let's go to the flies now. So we've built sort of a base here, and we'll sort of connect the dots over the next little bit of the conversation. Let's talk a little bit about flies and sleep and what was already known about the neurobiology here. So it sounded like you already knew that you had these very important sleep-inducing neurons which were sufficient to make flies sleep. You can turn them on and make flies sleep. Talk a little bit about the circuitry and the cells here that we already knew about that were really important for sleep in flies.
So the neurons that we studied are cells that project to a particular layer or two layers of the dorsal fan-shaped body in the central complex.